Hello guys.
Hope you guys enjoy our video.
2 hours later.
Hey Giri, what's up? How are you today?
Yeah, I'm so good.
Actually, I want to ask you a question because yesterday I have learned about chapter 7 in biology, which is DNA recombinant. But I didn't understand about the certain terms in that chapter. Can you explain it for me?
Yes. What a coincidence! Since I just check it on the internet and search some information about it. So, definition recombinant DNA is a form of DNA constructed in the laboratory. It is generated by transferring selected piece of DNA from one organism to the another. So, anything else?
What if we go to the cafe for some drink?
Yeah, why not?
A few moments later.
You know what? I think biology is quite difficult for us, since we have to study a lot and to understand the certain terms, and it is quite difficult.
Yeah, I agree with your opinion.
Okay, let's go. So Dani, since I know the definition of the recombinant DNA, so I want to ask you about the importance the recombinant DNA technology. So can you explain about it?
Yeah, sure. The importance of this DNA recombinant is the genetic engineering is used for many different purposes in research, medicine, agriculture, and industry. It is important because it enables the creation of multiple copies of genes and the insertion of foreign genes into other organisms.
Oh, okay. So I understand about it.
A few moments later.
Shakirin, even though you have explained about the definition, can you give some examples about the DNA recombinant?
Yes, I sure that you don't understand about the topics, since I only explained about the definition and the importance. So I want to give you two example about it. So first example is production of vaccines and protein therapies such as human insulin, interference, and human growth hormone. It is also used to produce clotting factors for treating hemophilia. Hemophilia. So, do you understand about it? Just about the production of vaccines, human insulin, interference, and other else.
Oh, I get it.
Alright,
Thank you for the information.
no problem.
A few inches later.
Hey guys, what are you doing here?
We heard that you guys talk about recombinant DNA technology.
So, shall we join you guys?
Yes, of course. Take a
Thank
seat.
you.
Careful.
Thank you.
So, we just discussed about the definitions, importance, and example of the recombinant DNA technology. We don't discuss about the tools and the steps yet.
Oh, I know the truth. There are some 5 tools in recombinant DNA. The first one is target DNA. The second one is restriction enzyme. The third one is DNA cloning vector. And the fourth one is modify enzyme. And the last one is process.
Oh, I know about the target DNA. Let's check it out. Target DNA. Sequence of DNA containing gene of interest. Example: gene encode for protein which can dissolve blood clots. Used in heart attack therapy. Do you guys understand about the target DNA?
Yes.
It's easy, right?
Yes. So, who want to explain about restriction enzyme and DNA cloning vector?
Me.
Yes, explain.
Let's check it out. Restriction enzyme. Enzyme which cut DNA molecules at a restriction site and produce short restriction fragments.
Restriction site is the short base pair sequence of 4 to 8 base pair recognized by a particular restriction enzyme. Restriction site contain palindromic where the same sequence of bases is found on both DNA strands but in opposite direction. There are two types of cut: sticky end and blunt end. Examples of restriction enzyme. First, EcoR1. It produced by E. coli recognize the base sequence of 5' to 3'. It makes staggered cuts within the sequence. It produces sticky ends, which is single-stranded ends. Second is SMA1. It produced by Serratia massacens. It recognizes the base sequence of 5' to 3'. The cut is made between the adjacent C and G. It cuts straight across the double helix. It produces blunt ends. DNA cloning vector. DNA molecule which can carry foreign DNA fragment into a cell and replicates in that cells. The characteristic: it able to accept foreign DNA in multiple cloning sites. It able to replicate freely in the host cell. It also possesses selectable genetic marker which is resistant to antibiotics. Example: ampicillin resistant and laxy. Examples of cloning vector: plasmid, which is DNA molecule that can carry foreign DNA into a host cell and replicate there. The capacity to carry insertion size is 1 to 10 kilobase. Second, bacteriophage. It is a virus that infects bacteria. A capacity to carry insertion size is 8 to 20 kilobase. Third, cosmid. It is a combination of plasmid and cosgene or bacteriophage. The capacity is 35 to 50 kilobase. Last, YAC. It is the fragment of DNA from human genome inserted into a yeast cell. The capacity is 100 to 1000 kilobase.
So you guys got that point?
Yes.
Good.
So how about another tutus?
Modifying enzyme. The first one is DNA ligase. DNA ligase join the foreign DNA fragment containing gene of interest and a bacterial plasmid. This is by catalyzing the formation of phosphodiester bond to form recombinant DNA. The second one is Taq polymerase. Taq polymerase is a heat-stable type of DNA polymerase that purifies from hot spring bacteria, which is Thermus aquaticus. It used in polymerase chain reaction, which is PCR, and have the same function as DNA polymerase. Taq polymerase also act along with a supply of 4 nucleotides in PCR process. It will copy the rest of the fragment as it will replicate in DNA beginning from primer. Host cell. Host cell is a cell which receives foreign DNA and allows the cloning vector of recombinant DNA. The example is genetically engineered E. coli. The characteristics is able to receive recombinant DNA, through transformation process. It also able to maintain the structure of recombinant DNA from one generation to another. It also able to amplify the gene product from recombinant DNA. Bacteria is the most commonly used as a host cell. This is because DNA can easily isolate from or introduce into a bacterium. Bacterial culture grows quickly, rapidly replicating any foreign gene they carry. The example is E. coli.
I think that's all the tools that used in the DNA recombinant technology.
Guys,
Yeah?
what time is right now?
It's 5:45.
I think we should stop our discussion right now. Shall we continue tonight?
Yeah.
Sure.
Yeah, we are.
Okay.
Of course.
Okay, bye.
Bye.
Bye, guys.
Bye.
Later.
Step in gene cloning.
12 seconds later.
Amy.
Step in gene cloning. Isolation of gene. Plasmid from bacterial cell is isolated. Gene of interest from DNA donor is isolated too. Cleave and cut. DNA donor and plasmid bacterial is cut and cleaved using compatible restriction enzyme or restriction endonuclease at restriction site in LEGZ gene, such as EcoR1 enzyme will cut 5' to 3' GATTC sequence at restriction site, producing complementary sticky end. Insertion. Gene of interest are inserted into the plasmid to form recombinant DNA are joined by DNA ligase.
Transformation and amplification. The recombinant DNA then inserted into the bacteria through transformation process that allow to multiply. As the bacteria reproduce, the recombinant DNA is also replicated. Screening. Screening is the process to identify host cells that carry gene of interest. Example, blue-white screening. Observation. Bacteria containing recombinant plasmid is white clonies. Bacteria containing non-recombinant plasmid is blue clonies.
Thank you for watching our video.
That's all from us. Bye.
Wait. Don't forget to like and subscribe our video.
Bye.